We are happy to welcome back to our studios the University of Western Ontario Professor of Virology, Yong Kang. It is wonderful to have you back in that chair.
Thank you very much for inviting me back.
You visited us a few years ago, and
Yes.
we're talking at that time about what you saw as potential.
Right.
It's more than potential today, but before we do that, let's just show a couple of maps to show some background for what we're going to talk about here. HIV, the prevalence of HIV in the world today. Globally, there are 34 million people living with HIV. There were 2.6 million new infections in 2009. And if we take a look at this second map, the worst cases, of course, are in Africa, where in some southern regions, as the dark coloring suggests, as much as a quarter of the population lives with HIV. However, here was the news back in December. The first and only preventative HIV vaccine based on a genetically modified killed whole virus has received approval by the United States Food and Drug Administration to start human clinical trials. Developed by Dr. Yong Kang and his team at the University of Western Ontario with the support of Sumagen Canada, the vaccine, SAV001, holds tremendous promise, having already proven to stimulate strong immune responses in preliminary toxicology tests with no adverse effects or safety risks. It is the only HIV vaccine currently under development in Canada and one of only a few in the world. So first of all, congratulations.
Thank you.
Second of all, how did you get from where you were when you last visited us to where you are today?
So we learned from the experience that others have and also others have published, and that the many different strategies that people have used. However, none of those strategies, unfortunately, worked out to prevent HIV infection. So we took the conventional wisdom, that is the kill the whole virus vaccine strategy works for so many other viral diseases. So why not HIV? So we have produced HIV and then inactivated by chemical and radiation and used as a whole, kill the whole HIV vaccine. But this vaccine, the virus that we have used are genetically modified so that it is not that dangerous and also produces large quantities when we infect the cells. So we have modified some genes of the HIV and then produced large quantities and inactivated, and now it's ready for human clinical trials.
Ready for human tests, but have you been testing it on animals up until now?
It's small animals as well as non-human primates.
Non-human primates. And how does it actually work?
Well, when we immunize a foreign body substance such as proteins, and this is, in this case, is a virus. Inactivated virus gets into the body of the animal or human, then our body reacts to this foreign substance and they make antibodies. And these antibodies then recognize invading viruses and neutralize, and thereby you can have a protection.
And has the problem in the past been that you've not been able to create antibodies strong enough to destroy HIV, which is so powerful?
Well, the strategies that people have used were not the whole virus, but taking the part of the genes of HIV, put it into some carrier virus, such as adenovirus or poxvirus, and then put it into the human. And the expression of those proteins in those immunized individuals maybe was not strong enough to get strong enough immune responses.
I see. Now, as we said earlier, yours is the only one in Canada so far showing success, but there are others in the world that are on it as well. Are they taking the same approach as you are, as far as you know?
Well, as far as I know, we are the only one who is using this approach, and there are many other approaches. In fact, there are about 4 different approaches to make vaccines against HIV. The one is, of course, the genetic vaccine known as a DNA vaccine. And then there's a vaccine which uses the viruses which doesn't cause any disease, but you can put HIV genes, part of it, into these the disease-causing viruses and make a recombinant virus and use that as a vaccine. So that in the past, there have been 2 companies used those strategies, and they used adenovirus and the poxviruses, and they did not produce strong enough immune responses.
Not strong enough. And any of the other ones that are happening elsewhere in the world, are they at the stage where you are in as much as they're ready for human trials as well?
There are other vaccines which are in human trials, phase 1 and phase 2, but none of those trials uses the same strategy as we use.
Okay, you mentioned earlier again that you've done this on small animals, small primates, and so on, in the testing phase. What is it that you actually see when you do it at that stage?
When we immunize those animals, then when we take out the blood of these immunized animals, and we look at the antibodies in the plasma, and that antibodies can recognize the viral-specific proteins. So we have techniques to identify whether or not there is antibody against those proteins.
And what you see under the microscope is that the antibodies are attacking.
No,
and
not in the microscope, but we can do what we call Western blots, so that we run the gels and then let it react with antibodies coming from those immunized animals, and then they show colorimetric the signals, so we can look at whether or not that there is antibody recognizing viral-specific proteins.
Okay. Because it's worked in animals,
Yes.
can you reasonably conclude that it'll probably work in humans as well?
That's what we assume, yes, because the human body reacts to a foreign substance such as the vaccines like animals do.
And what has to happen in order for this vaccine to prove to be viable?
There are three different phases, phase 1, 2, and 3. Phase 1, we look at the safety of this vaccine and in humans. So that's what we have approval for from US FDA. And we look at whether or not this vaccine is totally safe in human, and that's very important. Once we prove that it is safe in human, it is safe in animals. We have already tested in animals, but make sure that it is also safe in human. And once it's safe in human, then we go into the phase 2 human clinical trial. And that requires about 600 people and volunteers, and where we are looking at immune responses to these vaccines. So we immunize these people and look at the antibody production in these people. And once we demonstrate that they do produce antibodies against the viral-specific proteins, then we go into the efficacy test. And that is a phase 3 human clinical trial. And there we have to recruit approximately 6,000 volunteers for that efficacy test. And that test will tell us whether or not this vaccine is effective in preventing HIV infection.
Okay, let me go back to phase 2.
Yes.
You need 600 people who
Yes.
are prepared to be guinea pigs, really, right?
Well, they volunteer. In fact, we get a lot of requests, and I'm inundated with the requests, the volunteers.
So you don't think it'll be a problem finding the people?
No, there haven't been any problems for other trials, and that actually people can recruit the people, those high-risk group people, without any difficulties.
Now, do you need 600 people who have HIV?
No. This
No?
is the phase 1 human clinical trial. We are looking at about 40 people, and that's HIV positive but healthy individuals who has not developed AIDS yet.
That's it. That's
And
phase
that's
1.
for the phase 1, and that's just to look at the safety of it.
Okay.
And then phase 2 and phase 3, we need naive people who are not infected with HIV yet.
You need what kind of people?
Naive, we call it.
Naive?
Yes, they are not infected yet. This is the medical terms that we use, those people who are not infected, but at the high risk.
Okay, and then phase 3 is 6,000 volunteers.
Yes.
And what kind of people would be eligible for that?
There will be people from the high-risk group, and these include the hemophiliacs who need multiple blood transfusions, and there's injection drug users, and they are vulnerable for infection, and also these commercial sex workers, and the gay people with multiple sexual partners. And those people we categorize as high-risk group, and so that we will recruit those people for the phase 3 human clinical trials.
And again, because I don't know, is it tough to get 6,000 people from that
No,
group?
I don't think so. No.
Do you pay them?
Well, we have to pay their expenses to come in for their checkups because we have to have follow-up studies.
Oh, I see. Yeah. Okay. Now, this is, again, what's going on in Canada. I mean, you're in a race, right, with the rest of the world in some respects, right?
Well,
do you know what's
in a way,
going on
yes.
elsewhere?
Well, we do. We do know that how many phase 1 and phase 2 human clinical trials are going on. And those trials are using the similar strategy which have been tried in the past, and they were not successful. So hopefully that this strategy will work. And because it works for other viral diseases, such as hepatitis A, rabies, flu, poliovirus, like a Salk vaccine, and Japanese encephalitis, and so on, and the flu viruses. These are all kill-the-whole virus vaccines. In fact, in addition, there are about 16 animal vaccines using the same strategy. It works. So hopefully that this will work for HIV.
Let me take you sort of many years down the road.
Yes.
Would the idea be
Yes.
that, you know, kids when they go in to get their mumps and rubella and measles and chickenpox and all those other shots,
Right.
this goes into the mix? Is that the idea?
No, this has to be independent because the childhood viral diseases such as polio, measles, mumps, rubella, and so on, and we immunize them when they are in the adolescent or very young age, and to make sure that these children are really protected from these common diseases. And this, once it's approved, and once it's approved that it is effective vaccine, I guess any healthcare workers and anybody who has any kind of risk will take this vaccine.
But that's what I'm wondering. If
Yes.
I mean, we're at a stage right now, for example, I think in the last year or two,
Yes.
the federal government said HPV virus
Yes.
vaccine,
Yes.
everybody can get it.
Yes.
Do you see a day when everybody will be getting this HIV vaccine, or just those who are sort of in high-risk groups?
Well, this will be voluntary. I don't think one can ask everybody to get these vaccines. This will be voluntary basis. But anybody who thinks that they are at risk, then they may be immunized, because this will be, for sure, it will be safe and trigger immune responses which can protect HIV infection. So there's nothing to lose.
And again, for the record, do you want to say which groups are considered to be at high risk of contracting HIV?
Well, I had mentioned that there are three of the four different categories. It's hemophiliacs who needs multiple blood transfusion, and the injection drug users, the commercial sex workers, and then also those people, the gay people having multiple sexual partners. They are at the high risk group.
Unsafe?
Unsafe.
Unsafe
Yes.
sexual partners.
That's correct.
Yeah. A couple of business questions here, if I can.
Yes.
Here's intellectual property question number one.
Right.
Who owns the patent on what you're doing?
The University of Western Ontario has the patent, and in fact, I, as a faculty member at the university, we have in the collective agreement that any invention that faculty member creates, that property belongs to the university.
So if this does become huge,
Yes.
do you get rich?
University will get some royalties from the company, and then the university will share with the developers, including myself.
Okay. And I mentioned again at the very beginning, with the support of Sumagen Canada. Who are they?
Well, Sumagen Canada is a subsidiary company of a Korean company, biotechnology company, Sumagen, and it's established about 5 years ago to support our research. And in fact, we do need the office workers to handle all the documentations for the approval processes of US FDA.
How much money do you think you've spent so far trying to get to this point?
So far, they must have spent approximately $20 million.
20 million?
Yes.
It's called SAV001. What does that mean?
Sumagen AIDS vaccine #1.
Gotcha. Okay. Are you guys going to come up with a better name for it by the time it's all said and done?
Well, so far, we've been using that SAV001 as this first trial vaccine.
I see. But I mean, somewhere down the road, would you want it to be called like the Kang vaccine or something like that?
No, I don't want to put my name on it.
You don't want? No? I mean, we've got the Salk vaccine, right?
Yes,
After
of
Jonas
course,
Salk. So you
yes.
don't want the Kang vaccine?
Well, Salk vaccine is, I mean, there are two Salk, the vaccines for polio. The killed whole virus vaccine is Salk vaccine, and live virus vaccine is the Savins vaccine, right?
Okay.
So, but I don't want to put my name on it.
I understand. I understand. God forbid,
Yes.
but what if it doesn't work?
Well, God only knows. We know that it will trigger immune responses and will make antibodies and so on. Whether those immune responses are protective immune responses We don't know. Because there's no animal model to test the efficacy of the HIV vaccine in any known animals. And that's why its process is so long. It takes long because we can only test in human. So we know that up to the phase 2, we have no problem. We will have, it is totally safe, we know that. And then we will have a good immune responses in human, we know that. We can extrapolate that from the animal studies. But then we do not know whether this will be protective vaccine against HIV infection. That needs to be seen.
How confident are you that you're almost there?
Well, I'll leave that the confidence up to God, and the God will tell us whether we are successful or not. But certainly, we are trying our best, and hopefully that it will work.
Yong Kang, we're always grateful for your visits here at TVO. Good luck with your work, and thanks so much for visiting us.
Thank you very much for asking me to come.
Support Ontario's public television. Donate at tvo.org.